our biological terms related to genetics. We have alleles which are said to be dominant. Those dominant alleles will be represented by the capital letters. Here we are making an example of tallness. Tallness is dominant over shortness. That is the example. Where we have double T, T and T, which is representing the dominant alleles. These dominant alleles means that as they are two, they means that there is a diploid number of chromosomes. There is one allele from the maternal and another allele from the paternal origin. You remember that when we're doing the meiosis. And the recessive alleles, another term. When alleles are said to be recessive, is because they are masked or they are dominated by the dominant allele. They are masked. They don't show up in a phenotype of an organism. Here we have these recessive alleles which are represented by the small letter T's. This indicates that when there is a fertilization taking place in two individuals, one is tall, another one is short. The short characteristic will not show up, but the individual or the offspring will be tall, like taking the characteristic of the dominant alleles. In a homozygous condition, homo means the same. It means that both alleles are similar to each other. Then we have another one, which is the heterozygous genotype. In a heterozygous genotype, the alleles for a gene are not the same. We have capital letter T and we have small letter t that indicate that it's a heterozygous condition. And when we look at this phenotype, or the phenotype which is made by this genotype, the individual will be tall because the recessive allele is dominated by the dominant allele. The person will be tall. And we also have another term, monohybrid cross. In a monohybrid cross, mono means one. So we are investigating here the inheritance of one characteristic. It's one characteristic which is involved. The example of this monohybrid cross, we are going to make use of the same example that we have been using, the height, but the dominant characteristic here is tallness. It's tall times short. So the characteristic investigated here is the height. And we have another terms, which is the types of dominance. Types of dominance. So in these types of dominance, we get this information about it from Gregor Mendel, which is the father of genetics. We have three types of dominance here, which is a complete dominance, the second one is the incomplete dominance. The third one is the co-dominance. The third one is the co-dominance. Yes, in a complete dominance, if we make use of an example of a red times white flower, after fertilization, the offspring will all be red in color. because it's a complete dominance. It's a purebred red and a purebred white, or it's homozygous red and homozygous white, as we did the examples before when we were talking about these terms. This is a homozygous genotype. It's represented by 2 capital letters. And we also had the recessive homozygous, which is represented by the small letter t's. So this means that in a complete dominance, we'll be having the same thing. We'll be having the capital letters. Let me do the example of this complete dominance. Dominance will be having the phenotype, which is the external appearance of an organism. We'll be having the phenotype, which is red times white flower. Their genotypes will be homozygous red times homozygous white. We make use of R in both of them because according to the law of dominance, it says in genetics, the dominant characteristics, in a dominant characteristic, we take the first letter as the alleles to be used. That's why we are using 2 capital letter R's and 2 small letter R. We don't use W because it's a complete dominance. We make use of the R's, which is the dominant allele. If we can take a look at this, at the examples which are used here about the types of dominance. We have these 3 types. In this codominance, complete dominance, we are seeing that we may have incomplete dominance in a homozygous condition where we are having homozygous dominant and the homozygous recessive, as well as heterozygous condition. And in this case, you will see how this genetic cross will be happening, how the characteristics will be transferred from parents to the offspring. We are going to do it later. Then let us go to the incomplete dominance. In the incomplete dominance, neither of the 2 is dominant over the other. But when they are reproducing, they will produce a 3rd phenotype, which is different from both parents. The example of this, if we use the red flower and the white flower, after fertilization, the offspring or the flowers produced will be having pink color. Pink color is different from the color of the parents' flowers. They were red and white, but the offspring of the flower is pink, which is totally different from parents. That is the incomplete dominance. Thirdly, we have a codominance. In a codominance, both of them are equally dominant. Neither of them is dominant over the other. We have the offspring having the characteristics of both parents equally displayed in an offspring. Again, if we take an example of a flower, a red flower, homozygous red flower, and homozygous white flower, when they are fusing, the offspring of a flower produced will be having the patches of red and white, like this one we have outside at the beginning of our chapter. There are patches of red and white showing that neither of the parents were dominant over the other. That is what we are going to get. And in codominance and incomplete dominance, both the genotypes of the flowers are represented by the capital letters in their genotypes. But in a complete dominance, we were having 2 contrasting characteristics. The red flower as dominant gene having represented by the capital letter R, and the white flower as a recessive gene represented by the small letter r. Yes, after this I'm going to give you a task in page 75 of our textbook, understanding, not page 75, on page 77, activity 5.4. In page 74 we have activity 5.3. Remember, the more you do these activities, it will be easier for you to remember. After doing this, I'm going to explain to you about the Mendels Law concerning this genetics. I know at the beginning there is a lot of, you have to listen a lot, but it's important. Please, you have to understand all this. According to Gregor Mendel, who was awarded the name of father of genetics, that scientist was very concerned about how organisms are having characteristics similar to their parents, or they have those differences among them. He observed the plants, observed the animals, as well as human beings. Then in his investigation, he used pea plants, uphizi. He used pea plants where he investigated the height, the size, and the shape. That was his concern. He used the law, he came up with the law of dominance, the law of segregation, and the law of independent assortment. In his investigation, Mendel used the pea plant in order to investigate how the height is transmitted from parent plants to the offspring. He used a homozygous tall plant and a homozygous short plant in order to get the features that he needed. I am going to write this investigation. In his experiment, before we start this genetic cross, I think it's important to make use of the format, to inform you with the format that is used when we are doing genetic crossing. So it's very important that we do that. The Mendels Law first. The Mendels Law. The first one we said it's a law of dominance. In this law of dominance, he says that dominant allele will always dominate the recessive allele. That is what he says. Dominant allele always dominate or mask the recessive allele. The recessive allele, which means that as we spoke about the heterozygous genotype, in this case there are two different alleles. One allele is for tallness, the second one is for shortness. But when this person is viewed externally, the person will be tall. So that is the law of dominance. In any other condition, it can be the color, where the light complexion is dominant over the dark complexion. Just look at your family members. you may find that the father is bright in complexion and the mother is dark in complexion. But when you are looking at the children they have, you find that all children are having the bright color. That means the bright color is dominant over the dark color. It can be a vice versa. It can be anything. Sometimes it can be the height or the eye color. That is the dominant. Wherever they are contrasting characteristic, dominant characteristic will always dominate the recessive characteristic. Okay, and another one is the law of segregation. The law of segregation. The law of segregation is about meiosis. It is about meiosis. We've been talking about this. If you look again at this inheritance under the complete dominance, we find that we have two alleles, the capital letter allele and the small letter allele. So this indicates that it's a diploid condition. If we talk about the chromosomes, we said previously in human there are 46 chromosomes. When meiosis is taking place, those chromosomes are halved and become 23 chromosomes. Even here, when the law of segregation is applied, that means meiosis will take place and meiosis will end up giving the gamete with one T capital letter and another T will be small letter. That would be the gamete. In the case when we're talking about meiosis, we said after meiosis or during the formation of gamete, each gamete will have 23 chromosomes, which is the half chromosome number from the parent cell. The sperm cell will have 23, the egg cell will have 23. That is the haploid chromosome number. But when this sperm cell and egg cell fused together during fertilization, they will result in a diploid zygote, which is having 46 chromosomes. That is what we are talking about here, about the law of segregation. We are saying the genotype segregate, they separate from being diploid into haploid. That is meiosis that we have done during the topic meiosis, and when we are talking about the human reproduction. And we also have another one, another term here, which is the law of independent assortment. The law of independent assortment. That is the third law of Mendel. Alright, in this law, Gregor Mendel is relating to the second filial generation. As we are having our parents, in our parents we are the first generation. The children that we are going to have will form the second generation of our parents. So this law of independent assortment usually applied during the second generation, where we found maybe the offspring are all heterozygous. In that heterozygous condition, we find that when the offspring are grown up, are becoming parents, when meiosis is taking place to form the gamete, we'll find that the gamete will sort themselves anyhow. As we said during human reproduction, there is a chance fertilization. No one knows which sperm cell will be able to fertilize the egg. No one knows even during meiosis which gamete or which chromosome are going to be the first one during metaphase. So that is the random assortment. Even during anaphase, the way in anaphase 2, the way the chromosomes are pulled to opposite poles, it's just okay randomly. So that is also showing a random or independent assortment of chromosomes. So those are the findings of Gregor Mendel, who is the father of genetics. I'll make the format so that we'll be able to do the genetic crossing. When Gregor Mendel was doing his experiment, he used two ways of finding how inheritance is taking place. So those two forms, or those two formats, the two formats of genetic cross. Two formats of genetic crossing. We have a diagrammatic format, diagrammatic illustration. That is the first one, and I like this one very much because it makes you easy to understand. And the second one is a Punnett square. I'm just going to write these formats first as they are, then we will engage in doing the examples. The format is the first one, diagrammatic illustration. It says we must have P1. P1 stands for parent number 1. And in parent number 1, what do we see in you? I'm just seeing the individuals, but what do I see? I see a tall man, and a short woman. So these are two individuals that I am seeing. So this is phenotype. These are phenotype, P1 phenotype. I'm writing small letters now. P1 phenotype, we have a tall man and a short man. And after the phenotype, we can then look at the genotype. But the genotype is not visible to us. Genotype is informed by the phenotype is informed by the genotype. As we say, we have a tall man and a short woman. So when we are doing the genotype in letters, it means we have two capital letters for tallness and we have small letter t's for shortness. This is a genotype. These genotypes are diploid. One allele is from the mother of this man, and one allele is from the father of this man. Even here we have two alleles. One allele is from the mother, one allele is from the father of this woman. In an individual, there is a mother and a father because of the chromosomes that we carry in our bodies. So this means that these genotypes are diploid. As they are diploid, meiosis have to take place in order to form gametes. Meiosis will take place and it will result in the formation of gametes. Gametes are sperm cells and egg cells. So, as meiosis have taken place, now we will have capital letter T separated by a comma and another capital letter T. This means now this is 23 and this one is also 23. They are now haploid because of the comma. If you don't want to use a comma, you may circle that allele. Okay? Now let us look at the genotype of the mother. We'll have small letter T separated by a comma. This one also is haploid. This one is haploid, as they are separated. When does meiosis start to take place in human body? It starts to take place during the puberty stage, when menstruation begins in the females, in the young girls, and when the males also have that wet dream. That is showing that it's the beginning now of being a matured man. When this have taken place, that means now fertilization may take place. If these people may engage on sexual intercourse without using the contraceptives, even if the girl is 12 years, even if the boy is 13 years, the girl may fall pregnant if that have taken place during that time when the egg is ready. You remember when we were talking about this during the human reproduction. So when fertilization is taking place, we will have isizukulwane sokuqala, the F1 genotype. The genetic makeup. I'm going to make use of the lines. I'll draw them so that you see how we do it. The first line will start from the first T to the part where written genotype. It must be the first line, and the second line will be from the mother first allele, joining this one. So it means now this sperm cell has fertilized this egg. The offspring will be having the genotype, which is capital letter T and small letter t. Again, from this first allele of a father, we fuse with the second allele of a mother. We again get capital letter T and a small letter t. This is the second child. And we move to the second allele of a father. We fuse the second allele of a father and the first allele of a mother. We get the capital letter T, the small letter t. We have a third child. The last one now, starting with the second allele from the father, capital letter T and a small letter t, which is the first one of the mother. We have the capital letter T and small letter. Now this one. We have to move to the second allele now from the mother. So this one is cancelled. It means we have this capital letter, second capital letter of the father, and the second small letter of the mother. What do we have? The phenotype we have in this one. The phenotype we have is that all offspring are tall. This is a complete dominance, meaning that the dominant allele have dominated the recessive allele. That is why we have a tall children. We have tall children, all of them. So, in terms of the percentage of the phenotype of these children, we say they are 100% tall. So those are the questions which are asked. If you look at the mind the gap, the mind the gap is also showing us this template where nothing is written on it. In my template or my format, I even wrote everything. Here is it.